Hello and welcome to the Beyond Biotech podcast number 100, a milestone. I'm Jim Cornell and this is the weekly podcast from Labiotech. This week we're talking about mushrooms. Well, kind of. Alpha-amanitin is a new payload being investigated for antibody-drug conjugates. Although alpha-amanitin is a deadly poison produced by the death cap mushroom, it could be directed against cancer because of its potent cytotoxic properties. To tell us all about antibody-drug conjugates and how alpha-amanitin works, we had a conversation with Heidelberg Pharma's CEO, Professor Andreas Pahl. I wonder if first you could give me a bit of background on Heidelberg Pharma.
Sure. So Heidelberg Pharma was founded back in 1999 when Boehringer Mannheim was sold and was a management buyout. So they pursued some old projects for some years, but that was not that successful. And then Heidelberg Pharma reinvented itself more than 10 years ago by a PhD student who joined then as a PhD the company, and they took an idea from his old professor. And basically, this was the start of the story going into the ADC business. So this was all around a new toxin. And the ADC technology became famous at the first wave at that time. And the idea was to use that toxin as a new payload for antibody-drug conjugates and to make this then usable for cancer therapy. So that's basically the story. And we developed this for a couple of years as a platform technology. And then a few years later, so in 2016, we also started our own drug development based on the platform technology we developed so far.
As we're going to be talking a bit about ADCs or antibody-drug conjugates, I wonder if you could explain a bit about what antibody-drug conjugates are and payloads and how they work.
Yeah, usually we call it as the combination of the best of two worlds. So everybody knows small molecules, the chemotherapeutics, which work very well in killing tumors, but they are not very specific because you know all these side effects, so hair loss, nausea. You have a very hard time as a patient getting such a small molecule. So they are good, but they are very unspecific. On the other end, you have the antibodies, which are highly specific in targeting certain structures, but they don't have any pharmacological or cytotoxic activity on its own. And the idea was to use the best of both worlds to conjugate them together, so have a small chemical linker, and use the high specificity of the antibodies to deliver the toxic payload to the tumor cell. So you can call it like a cruise missile. So the antibody is the missile, let's say, flying to the tumor cell, and then it's getting uptaken by the tumor cell and delivering its payload to kill finally the tumor cell. So that's the beauty of the ADCs. And the belief is that you have a better therapeutic window because as compared to a traditional chemotherapeutic agent, which is killing every cell, now you're delivering specifically the toxic payload to the tumor cell and have a much better therapeutic window for the patient.
We're hearing a lot more, I think, about ADCs lately. Why is it such a hot topic at the moment?
I believe it came in waves. So again, it was 15 years ago where there was a first wave with the approval of Kadcyla, and everybody was thinking, that's now the golden nugget. But then it turned out to be not that easy as it looked like in the first instance. So there were a lot of lessons learned, a little bit the valley of death in between. And then you had different payloads, which was also not optimal for ADCs. And a few years ago, the Ichizankyo came out with the topo-1, so topoisomerase 1 inhibitors based on the exatecan, which is a very new class of toxic payloads, and they delivered fantastic clinical data. So really a new wave of ADCs with fantastic clinical data. We've seen a lot of deals, and this was the next generation of ADCs, which was delivering good clinical data. And people realized for solid tumors, so CAR T cells are very hot, bispecifics are very hot, but they are mainly working in hematological tumors. They have not really brought into play for solid tumors. But Solid tumors, there's much bigger population to target. The market size is much bigger. So for solid tumors, the ADCs seems to be the hot modality. And having been to ESCO for a few weeks, you see so many ADC talks for a variety of solid tumors and delivering good clinical data. So this is really, let's say, in essence, why they are so hot for the current time.
Right. Is that why there's been a lot of interest from big pharma?
Sure. They are risk averse. They are waiting until they read good clinical data, so they don't go for a preclinical or phase 1 asset. But now they realize that it's working, and you probably have seen the big money that each is now getting from AZ, from Merck, multi-billion-dollar deals. A lot of early-stage companies are acquired. So now big pharma is jumping on the train, so they don't want to miss the train. No big pharma can afford, meanwhile, to say we are not in ADCs. So that's why it's really, let's say, getting so hot. Again, seeing the potential, and if you look at ESCO, every big pharma has now ADCs in the pipeline, and in particular for solid tumors.
Do you think that there'll be more FDA approvals in the near future in the area?
Yeah, definitely. You have seen the Daiichi programs coming. A lot of follow-up programs are coming. We will see a flood of new ADCs. Of course, what is also happening, so that's my personal view on this one, that there are quite a lot of ADCs coming, also a lot out of China. So China is, let's say, producing in a highly proliferative way ADCs. But the quality is maybe mixed. So there will be some failures in the clinical trials, and not every ADC will be a success on its own. So that's not true. It's not like baking bread. So there will be coming a lot of approvals, but not every ADC will be successful.
Yeah, for sure. What other ADC payloads are being used, and what can they be used to treat?
The beauty in theory of the ADCs is that just by selecting the antibody and the target for the antibody, you can address every tumor type. And as an example from our payload, the amanitin, we have never found any tumor cell which was resistant to the amanitin. So it's just the design of the ADC that you identify a suitable target, maybe for colorectal, ovarian, or whatsoever, and then use your payload. And what is also seen in the space, there's a lot of follow-up ADCs. So if an ADC on one target is approved, a dozen of companies jump on the same target with the same or different payloads. There are a lot of follow-up ADCs and not so much variety in the targets for using for different tumors. So this is maybe a limitation. And still, the variety of payloads is very limited, because if you look in the clinical trials, it's really dominated by either the tubulin inhibitors or the topoisomerase 1 inhibitors. Other payloads are really marginally, and it's very clear that the field needs more payloads, because if the tumor becomes resistant to one class of payload, you cannot treat with the same class of payload again, because the tumor is resistant. So you need to switch for different payloads in order to overcome the resistance of the patients. So there's a little bit, let's say, a duopole in the clinical trial setting.
Following on from that, when you are using ADCs, how do you avoid toxicity issues?
That's a challenge. So despite what I having said, let's say you have a targeted delivery to the tumor cell. It's not like mother's milk, how a CBO call it. You still have toxicities, and they're really, let's say, defined by the payload. Because if the payload kills the tumor cell, it's released from the dying cells, and it still needs to be eliminated from the body. So the old tubulin inhibitors are, let's say, moving around in the bone marrow, killing blood stem cells, doing, let's say, hemotoxicity. For the topoisomerase 1, there's a similar, let's say, heme toxicity, some kind of lung toxicity is observed. So you cannot really completely get rid of this one, and each payload class has a specific set of toxicities, and you need to manage these toxicities. You need to find ways in your ADC design to minimize, but you can't not completely avoid these payload-specific toxicities.
How important is the linker?
I get this question, so also I've been to the panel at ESCO. This is quite a frequent question. I have to say I'm biased, coming from a payload company. So my view is a little bit negative on this one because, again, in the end, the payload is released from the tumor cell, whatever linker it was used for conjugation. There are ideas around about, let's say, tumor-specific release of the payload. I haven't observed not so much advantages coming from this one. So again, I know there are linker companies, they probably have a different view on this one. My personal view, they are not the most important piece of the puzzle.
All right. What's alpha-amanitin?
Yeah, alpha-amanitin by nature is coming from a deadly mushroom. So it's called, in German, it's called the Grünknollenblätterpilz. In English, it's the green death cap mushroom. And it's still the most common source of death after eating the wrong mushrooms. It needs some skills to identify this one. And quite, let's say, 30 to 50% of the people ingesting a sufficient amount of the mushrooms are dying from liver failure and subsequent organ failure. So that's the amanitin, that's the poison, the toxin of that green death cap mushroom. And the idea, of course, in nature, he wants to avoid to be eaten by small animals. And it has a very unique mode of action. So it has been used in cell biology for years because it's a specific inhibitor of eukaryotic transcription. And the beauty for us, it's the only compound known in the world with that mode of action. And since the eukaryotic transcription is also very important to tumor cells, so if you deliver the amanitin to tumor cells, you immediately drive them into cell death and you destroy the tumor cell. So this was the idea in the beginning. But the amanitin itself is too toxic, so it was never used as a single agent, as a small molecule. And the idea, how I call it, was to harness the ADC technology and build the ADC technology around the amanitin and then make cancer drugs out of this one.
How are you able to make such a deadly toxin useful and not remain toxic?
Again, with the ADC, we detoxified the amanitin. So it's really that the tumor-specific delivery. And it turned out that finally you need only a rather small amount of the amanitin or a very small amount of the amanitin to kill the tumor cell. So to give you an idea, let's say if we give our ADC to the patient and the amanitin would drop off the antibody within a second in this patient, the amanitin dose is still too low to kill that human being. So we are far below the mushroom toxic dose because when people were coming to the hospital, you could, let's say, back calculate the amount of mushroom they have ingested and how much amanitin was. So it's very much known that, let's say, you have 0.1 milligram per kilogram is the LD50 for men for the amanitin. And we need a much lower dose for killing tumor cells because we are so specific by using our technology, conjugating this to the antibody, and again, a very low amount of the amanitin is needed when it is delivered to the tumor cell to kill the cell. So that's the final trick. So the secret sauce of Heidelberg Pharma.
And what kind of tumors are you using it for?
Yeah, we have developed of our pipeline. Again, we can address in theory every tumor type. Our first program we decided years ago to target multiple myeloma, where we are already in phase 1 in multiple myeloma patients. The next program will be targeting B-cell leukemias and lymphomas. And then we are going for the solid tumors for prostate and colorectal cancer. So that's the pipeline we are building up with the first 4 projects. But we're also partnering our programs and look for other companies using different antibodies to really maximize the commercial success of the platform as well.
Makes me think when a toxin is being used to treat something, how is that connection initially made between here we have a deadly mushroom and we have a disease? How do you make that connection?
The connection finally is that for every tumor disease, you need to find a marker, a target on these tumor cells. And these are very different for each tumor type. There are only a few, let's say, which are across solid tumors. And for example, for the multiple myeloma, because the myeloma comes from plasma cells which are in the bone marrow, and it was known that there's a molecular marker on these plasma cells. And then people found out in research years ago that there's a specific marker on the myeloma cells. And this is finally the connection. And this marker is different if you go to non-Hodgkin lymphoma. This is different for prostate cancer. So for that specific question, I believe the answer is that you need to identify a molecular marker on the surface of these tumor cells, which makes the connection. And then it's all about biochemistry, finding the antibody for that marker. So these are, let's say, biochemical and molecular biological methods, which basically do the trick. But the link, to answer your question, is the specific tumor marker, let's say. And this is one of the challenges. There is hardly any ideal tumor marker because every marker is to some extent expressed on healthy cells. So you need to find a huge expression, higher expression on tumor cells and whatsoever. It's also partly explaining the toxicity of the ADCs.
In practical terms, how is the amanitin delivered and how does it work in the body?
So finally, the ADC is, let's say, given intravenously. Not to say we have also are developing a subcutaneous delivery, but the standard delivery is the intravenous. It's then circulating in the bloodstream. In terms of a hematological tumor, it's very easy. Let's say the ADC looks, again, the antibody looks for the target structure on the cancer cells. For solid tumors, the ADC needs to diffuse out of the circulation and to find the tumor. And then as soon as the antibody recognizes the structure on the surface of the tumor cells, it binds to the structure. And then there is let's say, a cellular mechanism which is called endocytosis. So as soon as the antibody binds to the target on the surface of the tumor cells, it's a process called endocytosis. So it's, let's say, the membrane engulfs. So there's a small, let's say, hole in the membrane. And then this hole, let's say, is cut off of the membrane, and then a new structure, which is called a vesicle, is then endocytosed from the surface of the tumor cells. And then this is a cellular process where the content of this endocytosed antibody or ADC target antigen is destroyed in these endolyses, somehow they are called, and then finally the toxic payload is released because the structure is then also disassembled in the inside of the cell.
As far as the delivery is concerned, you mentioned that it's intravenous. How often does it have to be given, and is it something that works as a monotherapy or is it in conjunction with other treatments?
There's been a little bit of a tradition in the field where, let's say, it's given every 3 weeks. But there are, let's say, changes to this called standard regimen because there's a certain half-life to each biological drug and also other drugs on the market which are given in a weekly cycle or given for 2 weeks and then the pause of 1 week. So this is finally the art of clinical development. What is the best way to maintain the efficacy of the drug, but to avoid the toxicity as its best? The short answer is every 2, every 3 weeks, maybe every 4 weeks is given intravenously, but there are different regimens with regard to frequency where they have more frequent, but then at a lower dose. And usually in a phase 1 trial, it's given as a monotherapy most of the time because you want to understand your drug, the toxicities and the efficacy. And if you give it in combination, it's challenging to dissect what is the contribution of your new drug to the efficacy and the safety. But as soon as you have a safe dose, companies are thinking about combination therapy. So still ADCs are given in monotherapy, but as it's known from the world of chemotherapeutics, combination is still the key or standard regimens, let's say in cancer or combination treatments. And this is very specific and very individual for each ADC, for each tumor type, to look in the best form of treatment regimen for this ADC.
Could you tell me a bit about the clinical trials that you're doing, where you're at with those?
Yeah, as mentioned, HDP-101, how we call our first program, multiple myeloma. We have now the first 5 cohorts passed. The first 4 cohorts were mainly, let's say, you need to start with a rather low dose. We haven't seen so much in the first 4 cohorts. no safety signals, no efficacy, but one. So we had one patient in the third cohort which went on a stable disease. So you need to think of these patient population end-stage patients because they are passing all lines of known therapy. And sooner or later, they have become resistant to everything which is on the market. And then this is the population we see with our drug. And again, if we then, with a monotherapy of our drug, we see a patient in a stable disease, so he's not, let's say, progressing in his tumor disease for more than a year. Let's say, this was the first very good success of our drug because usually these patients have only weeks or months to survive. So this was the first hint of efficacy. And then with the fifth cohort, we have seen much more. So that's typically you escalate and you see then sometimes tox efficacy coming. And there they were very, very pleased to see that 3 out of 5 patients, which has been repeatedly dosed with our drug at the dose of the fifth cohort, we have seen objective responses. And objective responses are defined by a certain reduction of tumor cells in these tumor types. And we have seen partial remissions in these 3 out of 5 patients defined by international standards if you have more than 50% reduction in the tumor cells. So we were very pleased to see that in these 3 out of 5 patients, we have seen more than 50% reduction of the myeloma cells and with a very easily manageable safety profile. So we are also optimizing now the treatment regimen, which would then allow us to dose further higher so that we really see a little bit more room for escalating the dose to even improve the efficacy and the duration of response of these patients. But I believe in our view, it's the really clinical proof of concept that this new payload or a new toxin is working in the patients with a very good safety profile, and which is also very different from others and which can overcome resistance. So these patients we are seeing have seen 5, 9, even 15 prior lines of therapy, and they are still responding to our drug, which is a huge success for us.
Yeah, that's excellent. And you also recently received orphan drug designation for HDP-101. What are the next steps there?
Correct. So we were very pleased to see this. And the idea for us is, let's say, by the end of the year, that we have reached the end of the phase 1, and then we start the phase 2A trial, how it's called, so that the dose we identified then to be safe for the patient to roll out to a higher number of patients and to have a more robust efficacy signal in these patients. And then we have also a new biomarker for our payload where we can identify high-risk patients, which will, let's say, very likely to respond to our drug, which have a very poor prognosis. And with such a setting, you, let's say, may go to the FDA to ask for an accelerated approval. So the idea, again, with this phase 2A part and to turn these patient cohorts then to enrich the patients and turn this into registrational cohorts, and then to seek accelerated approval for this drug to really speed up the development for the first market product based on our technology.
Do you have any timelines on that yet, when you expect or hope to be able to?
Yeah, this is always very challenging again. So we will start with the phase 2A part next year, and then it's a little bit the length of recruitment and the discussions with the FDA. Whether this will take, let's say, 3, 4, or 5 years is a little bit difficult to predict. And obviously, multiple myeloma is a huge market, and we are, let's say, a relative small company. Sooner or later, we will probably seek for pharma partners who can join that enterprise, because that's probably not on us then to market and sell this drug to the myeloma patients.
As far as ADC payloads goes, where do you think that field is headed in the near future?
So I believe now we see the peak of the topo-1 inhibitors, because in my feeling, every known target has now an ADC program with a topo-1 on it. So this field is then the topo-1s has passed the peak. And the field is looking for new toxins with new mode of actions, where we believe fit very well in this one. And what we observe for other companies, for big pharma, they are really looking, scouting for new payloads with new mode of actions. But even from our, let's say, own experience, I have to say it's quite an enterprise to get a new toxin to work, because every payload is different, has different physical chemical properties, different mode of action. You need to learn about the toxicities, the best ADC design. So there will be huge effort now, is ongoing for these new payloads. And it's very hard to predict which will be the new star of the show. So again, we hope that amanitin will be one of the new stars. So that's where, let's say, our goal. We are working very hard. Probably others will come up soon with different modes of action.
You mentioned that kind of complexity. Are things like machine learning and artificial intelligence, are they making a difference?
Yeah, this is the buzzword of the year, of course. I believe what AI needs, it needs a huge database. And since there are hardly any successful payloads, so let's say if you have tubulin inhibitors and topo-1s, so I believe even AI cannot say with a very small database can generate something new. It's, let's say, maybe for identifying new targets, I see more room for this one to identify the best properties of the target, to look in the patients and the clinical data. So there I see more room for the AI, for new payloads. Again, I believe the amount of available payloads which needs to be fed into these AI programs for the machine learning, is much too small to generate new ones. But again, maybe there are surprises. I don't know, but this is my view on that one.
Is there anything else that you wanted to mention that we didn't talk about?
I believe a very, very good set of questions. Again, coming back from ESCO, there's a huge wave of ADCs coming. Concern is a little bit that the quality is very, very different, but there will be a huge variety of new options, especially for solid tumors for patients, especially tumors like pancreatic, colorectal, which there are not so many options for the patients. So I believe the ADCs will deliver very much in the future.
It's interesting how toxic compounds might be great tools against disease if used in a certain way. There are plenty of plants or parts of plants that are poisonous unless they're cooked, or only certain parts are edible, so clearly lots of opportunities to investigate how toxic compounds might be useful in disease treatment. Don't forget to check out the latest news and articles over at Labiotech.eu, and I hope wherever in the world you are, you have a great week ahead. Thanks for listening, and I hope you'll join us again next time for another Beyond Biotech.